people doing research here.
Mm-hmm.
We each have our own little section. This one happens to be mine.
Yeah. Closest to the door.
Yeah. Yeah. Very convenient. And I have all sorts of stuff here. To most people, it looks like a mess, but it's all organized just to how I think through things. Right now, I mean, one of the easy things to show you is, let's see, I have just plates of the bacteria growing.
Okay. So what is that?
It's called an LB plate. It's just a basic nutrient-rich medium that has, everything the bacteria need to survive. And what we do is it's called plating. We call them plates, plating. We plate the bacteria on there, and then they have some time to grow, and then they grow into these individual colonies, which can then be used for whatever I need to do with them. One example of this, come this way, we have a big room full of different cultures where we have everything incubating.
Like a big freezer. Oh, wow.
Yeah. Hi, Jose. So some of the cultures, like I can take the strains from the plates and put them into liquid media with different food sources, and then I can grow them that way. So that's one of the experiments that I'm working on.
Okay. It's like Dexter's lab in there.
Yeah. Yeah, it's all sorts of things. If you want to go in,
yeah, sure.
So,
tell me what I'm looking at here.
So we have a couple different shakers. This one is just a bunch of my cells growing on different types of food. They're just in there for a couple of days so I can see what it turns out to be at the end. Up here, this is one of our grad students' work. He's doing what's called a co-culture. So he's working to put two different bacteria to get them to live together in the same environment.
Okay.
And then we have a variety of just different liquid media and liquid cultures that are sitting there being warm and being shaken.
So what's this whole room called officially?
I call it the incubator room.
The incubator room. Okay. Okay.
Sorry, I have to think.
No, yeah, take your time, no worries.
So right now I'm doing incubation of some of the DNA with a digestion enzyme, which is going to basically cut it up into different pieces so I can see. I mean, you can't really see much in here, but it's just, this is one of our incubators, and, they sit in there for a couple of hours, basically just making sure that they get cut. This is one of the things I use all the time. It's for gel electrophoresis, which is used to separate a molecule by its size. So what I do is I make a gel, which is just kind of like Jell-O, but a little denser. And you put it in here and you run an electric current through there. And what the electric current will do is it pulls the molecule through the gel. And since the gel is porous, the molecules that are smallest can move through fastest. Kind of like if you imagine running through a field of people, right? If you're really, really small, you can run through much quicker. Where if you're a larger piece of DNA, it's going to be harder to get through. You're going to bump into more things. And so the bigger pieces end up toward the top of the gel, and the smaller ones are down at the bottom. And then we have a standard that we can compare to, so we know what size each of them are. And from there, I can actually cut them out of the gel and purify them so I can work with bits of DNA that I need to.
Okay.
Let's see, as far as that goes, it's most of the stuff. If you want, I can show you a couple pictures of like what it turns out after that.
Do you want to just tell us, just regarding the HHMI program, when this is done, what do you want to have accomplished?
Well, hopefully I will have gotten to a decent point in the project. There's, of course, always more things to do, more questions to ask. It's never ending. But I'm hoping that, I will have made some significant progress in my research, as well as, getting through the program it's a really good feeling to have, because you can, like I mentioned before, you have the ability to say, I did this research, I worked on this presentation, I get to the end of the summer, I presented it, I shared my work with everybody, and it's really a good way to feel accomplished about your research.
Perfect.